So today we're going to continue talking about skeletal muscle, but we're going to start talking about metabolism. So we're going to talk about where skeletal muscle gets energy, because that's really important to how we function, and especially during exercise, we'll see that there are different ways different muscle fibers are making ATP that influences how quickly you fatigue, how long your endurance is, which has real effects on your activities in the world. So as we go through this packet, so as we go through this packet, I've cut a couple things. I cut the section on muscle spindle fibers and Golgi tendon organs. So if there are slides missing as I go through compared to what you printed out, that's because I decided we would cut some content for time. So these last couple slides are from the final part of the previous packet that we didn't finish before the exam. And I'm covering them here just because they're going to be related to what we're talking about with the metabolism. So yeah, so we're not testing this part directly. It's just one of the things I'm about to say about metabolism in a couple minutes isn't going to make sense unless we remember this from physiology lab. So I want to make sure that we do remember this from physiology lab. Okay. So this is just a note about force generation and how that works in muscles. So remember, we have these different motor units, and they're of different sizes. So motor unit, we have one motor neuron innervating several different muscle fibers, and they all contract together because they're attached to the same motor neuron that has that same action potential. So very basic thing about muscles is the more motor, the more fibers we have contracting. Oh, okay. It looks like a bunch of people are messaging me that they don't have their exam displaying in the gradebook. It's all displaying in my gradebook, so I'll check why that's not visible to you. Actually, yeah, is that everybody?
There's a way to look at them. If you go to activities, quizzes, find the exam 3, and then look at submissions, that's how I've been able to see my score. It doesn't just show up like in your grade book.
Yeah, it's like calculated in like the running total of the grade book, but like there's not like a...
Oh, it's in a weird place somehow?
Yeah, like you can see it if you hit like the grade calculation, because I noticed that went up. That's how I checked.
Okay. Okay. Yeah. So people who are wondering, check there. I will fiddle with the settings after class and see if I can get it to show up in your normal spot, which hopefully I will be able to figure out from the student view. The student view for the professors is kind of not actually a real student view. Okay. Check the chat. That's a different chat. Okay. Yeah. So more motor fibers contracting, greater tension, greater muscle force. And we're going to recruit motor units in a specific order. So we're recruiting those muscle fibers attached to a specific motor neuron in an order. We're going to be using small motor units for delicate, careful movements, and we're going to be using large motor units for strength, because large motor units will have more fibers, stronger. And the order is just that we're going to be recruiting those small units first, right? So picking up a pencil, we're going to use those small ones. But when you're pushing something really hard, we're going to recruit small ones, then medium ones, then large ones. So that's basically the only takeaway I want you to get from these 3 slides at the beginning. So this is the picture that corresponds to that. You may have realized I prefer pictures to words sometimes. So this is just showing us that first we would recruit this, this motor unit here, this motor unit X. So this shows that us here on the graph as well. And then we'd get to Y, and then we'd get to Z. And this has to do with basically like how much of an action potential we need, how great that frequency has to be. Okay. And that will be related to our metabolism in a little bit. Okay, so when we're talking about our muscle metabolism, we want to be thinking about its metabolic demands, by which we really mean like how much ATP does it need to do all its cross-bridge cycling, to do all its contracting. We're going to relate that to different types of skeletal muscle, and we'll see that different fibers and different motor units are able to produce ATP in different ways with differing degrees of speed. So hopefully we remember something about this diagram on the right here. So here we have our cellular respiration, our flow, right? We start with glucose. We have glycolysis, which doesn't require oxygen, produces a little bit of ATP. Then we move into the mitochondria. We break down pyruvate, enter the Krebs cycle or the citric acid cycle or the TCA cycle, same difference. And then finally go into oxidative phosphorylation, where we produce a lot of ATP. Okay. So ATP, we're going to need it to do work with our muscle. And we are going to be able to use this process in our muscles, but different fibers are going to focus on different methods of ATP creation. Okay. So, right, we need that ATP in order to drive the cross-bridge cycle. Remember, there's that end point where we basically need to re-energize the myosin by hydrolyzing ATP. We need to break down that ATP in order to put myosin back into its high energy state so that it can shove our actin towards the center of the sarcomere again. So that's why we need this ATP. Okay. So we have three sources that we can use. I'm going to touch on points 2 and 3 first, because they should be familiar to us, and we'll jump back to point 1. So the one we've talked the most about, I should say, is number 2 here, the oxidative phosphorylation in the mitochondria. So that's where we had the electron transport chain. We had all those electron carriers, bringing those electrons to the electron transport chain. We were building up a hydrogen gradient between our 2 mitochondrial membranes, then letting it flow through the inner membrane into the matrix, spinning our ATP synthase, producing ATP. So that's the oxidative phosphorylation. This is this process that requires oxygen, because oxygen is our terminal electron acceptor. Okay. So that's one way we can get ATP, and that's pretty efficient, right? We get a lot of energy from our glucose that way. Our less efficient way of getting energy that we discussed when we were talking about respiration was anaerobic glycolysis. So remember right at the beginning here of this whole process, we break down glucose, we split it in half, we do this cleavage step, and we get ATP as we're splitting up that glucose into 2 parts. We remember that we only got 2 ATP on net, so this wasn't super efficient, but the useful thing about glycolysis is that we can run it in the absence of oxygen. So we could loop back, do fermentation, right? And produce lactic acid from our pyruvate, and then just kind of cycle through this glycolysis part without entering the rest of this pathway, so that we can keep producing ATP even if we don't have the oxygen to accept our electrons at the end. So we can't run the electron transport chain, so we can't do oxidative phosphorylation. Okay. So oxidative phosphorylation is great if we have O2, but otherwise you use glycolysis, we do fermentation, we produce lactic acid because it's less efficient, but we don't need the oxygen. Okay. The thing we haven't talked about yet is this first one, the phosphorylation of ADP by creatine phosphate. So we're going to go to the next slide to take a look at that. Okay. So in your normal cells, when you start out exercising, like right before you're exercising, right in the beginning phases, you start using the ATP that's in your cells. And you start to use it up kind of fast. You pretty quickly kind of burn through the ATP that was just kind of hanging around in that cell. But at this point you are exercising, right? So you still need ATP. And one of the first places you get it is from this molecule called creatine phosphate. When your muscle cell is at rest, we have this equation that's at equilibrium. You have a certain amount of creatine phosphate. You have a certain amount of ADP floating around in your cell. And you also have this molecule creatine and this molecule ATP floating around in the cell. So nothing in particular is happening on net with this reaction when you're at rest because it's in equilibrium. So it's moving to the right. We're converting creatine phosphate and ADP into creatine and ATP at the same rate as we're going backwards and creating creatine phosphate and ADP. Okay. But once you start to exercise and you start to lose ATP, right, because you're using it, it forces this equation to move to the right, right? We have briefly kind of touched on this law of mass action before, but I don't know that we've really emphasized it. It's more of a chemistry concept, right? But it's basically when we have a lot of buildup of something on one side of our equation, that's going to kind of propel us because we want to be at equilibrium. So we're gonna move our reversible reactions in that direction. So when we're using up this ATP, because we're starting to use more energy, we're starting to contract our muscle cells, suddenly we don't have that much ATP. So we're going to start making creatine. So we can use this creatine phosphate and our enzyme creatine kinase to convert ADP to ATP. And you only have a certain amount of creatine phosphate. There's not a ton of it. This is not going to power your cells for very long, but it will power your cells to the point that they're able to change things about their transporters on the outside of the cell in order to soak up, for example, more glucose to continue to use this energy then. So we think of this energy as kind of being on tap, because after your cell then starts producing ATP through its normal metabolic processes, so once you have plenty of ATP again, this process kind of stops because it goes back to equilibrium. It pushes this law of mass action in the opposite direction. So this is like our quick way to get ATP when we're suddenly running out of it. So you might have heard of creatine before. They sometimes sell like pre-workout powders, right, that involve creatine. And the idea behind those is to try and get you a little more of this kind of energy on tap. So they're often mixed together, or creatine phosphate with amino acids and other things that can help you build protein. That's the idea. Jury's out on what they actually do and whether it works, but this is the basis for that. Okay, so ATP stores are used kind of differently depending on the intensity of your exercises. So we're going to see different things happening with light or moderate activity versus super heavy intensity activity. So regardless, we are going to be able to chart what source of energy we're using as time goes on. So here we can see this chart, and we have time on the x-axis, and we have the energy that we're using from each of these sources on the y-axis. So as you start to exercise, as you start to do light exercise, first thing that happens is that you start to use up that ATP that's in your cell at rest. So this starts this conversion of creatine phosphate to creatine, giving us ATP. So that's what we see in these first couple of minutes, right? Is that we're using this creatine mechanism of producing ATP. As we move forward then, we start to do more glycolysis, right? So we start to do some glycolysis, and we also start to do oxidative phosphorylation. With light exercise, what we're mainly going to be doing overall, you can see as we've warmed up, right, by the time we're at minute, right, like 2, we're doing mostly oxidative phosphorylation. That's because with light exercise, we're able to oxygenate our tissues sufficiently that we are able to do oxidative phosphorylation. However, there is this kind of lag period in the middle here where we're trying to get enough glucose to the to these cells. We're trying to get enough oxygen to these cells. We have to start pumping our heart a little harder. So for a little bit here in the middle, we'll be relying on glycolysis. The thing about oxidative phosphorylation, right, is you have to keep your O2 supplies adequate here. So you have to breathe harder. You have to increase your heart rate, right? You need to breathe harder to get the oxygen in the blood, and then you also need to get that blood around faster to the muscles. So we're going to be increasing the heart rate. We're going to be increasing the power of those contractions. So we're going to be trying to force more blood through more quickly. We're also going to be dilating our blood vessels that go to our muscles, because as we'll see as we move into the cardiovascular system, when you increase the diameter of things, you basically increase the speed with which blood can get to those tissues. Okay. So this is what's happening with our light to moderate intensity. We're heading towards this oxidative phosphorylation. Oh, yeah, I should say we use glycogen while we have it, but we do run out of that as well, just like we run out of the creatine. After about 30 minutes, we've changed the cell membrane so that we have more proteins that are able to pull more glucose out of the blood. So we kind of adapt. This is going to be kind of for an endurance type of activity. We note the contrast between light, moderate intensity activity to heavy intensity activity. So heavy intensity activity is really mainly going to be using purely anaerobic glycolysis. So we do start out with our little sort of creatine bump, right at the beginning while we start to exercise. But then during the bulk of our activity, what we're doing is glycolysis, this less efficient process. So we're producing lactate and lactic acid buildup. They give us a burning kind of sensation in our muscles during this high intensity activity. We're only making about 2 ATP molecules per glucose, but the sort of benefit here is that we can have bigger muscle fibers, and we don't need to get them oxygen really because they're not using oxygen. So we're going to be able to make more powerful movements, mainly because we can make the fibers like so big that they would be hard to get oxygen all the way through into the mitochondria if we were trying to do oxidative phosphorylation. Right. And we're going to be thinking about light versus heavy intensity activity throughout these next couple of slides. So that's the basis for this stuff. But as we think about individual skeletal muscle fibers, we're going to classify them in a couple different ways. So we'll actually see that some muscle fibers are specialized for anaerobic glycolysis. Some muscle fibers are specialized for doing oxidative phosphorylation. So we're going to see some muscle fibers being specialized for not using oxygen. Some muscle fibers are going to be specialized for yes, using oxygen. So that's one way we're going to classify our muscle fibers. The other way we're going to classify them is based on kind of how fast they work. So we talked about the twitch at the end of our last unit. We're going to see that twitch happens more quickly in some muscle fibers and more slowly in others. So we're going to be thinking about the combination of kind of these two factors as we're looking at the function of an individual muscle fiber. Okay. Yep. So we're going to have fast and slow fibers. We're going to have oxidative and glycolytic fibers. Okay. This is our cross-bridge cycle. Hopefully this looks familiar. And I'm pulling this up because this kind of explains what's going on with this fast versus slow twitch. There are actually two kind of versions of myosin, and they differ in how fast they can hydrolyze ATP. So when we're saying slow, we're saying that it's a type of myosin that breaks down ATP slowly, So all this cross-bridge cycling happens more slowly. So our contraction is also going to happen more slowly. So it's just helpful for me to see this, to kind of get into my mind what we're talking about when we're talking about slow versus fast. Okay. So we're going to be contrasting these fast twitch and these slow twitch fibers. This is dependent on that rate of the myosin ATP-ACE activity, right? So the hydrolyzing ATP breaking it down to ADP and that phosphate ion to get that energy out of that bond. So our fast twitch fibers are able to do the cross-bridge cycling more quickly because they have this fast myosin. And these fibers are also going to relax more quickly. Okay. In contrast, our slow twitch fibers are going to cross-bridge cycle more slowly because they have this slow form of myosin, but it also means they're going to take longer to relax. So in different parts of the body, so in different types of muscles, we're going to see different balances in these fast and slow twitches. So here again, we have time on the x-axis, and on the y-axis here we have tension. So what we're looking at is basically how fast it takes for each of these muscles to get to its peak tension. So its peak tension is like when it's contracted as much as it can contract. When we looked at, you may or may not remember, but the graph of a twitch, we had this latent period, we had this contraction phase, and we had relaxation at the end of that. So these are all sort of altered versions of that graph of the twitch that we had in our previous unit. So we have a latent period while we're sending the action potential from our neuron down our sarcolemma, down our T-tubules, starting our cross-bridge cycling. And then our fast twitch and slow twitch fibers are going to start contracting, right? So our fast twitch extraocular muscles are contracting. Our gastrocnemius is kind of intermediate in speed. It's also started contracting, but we can see that to get to this peak tension, it's getting there kind of slowly. Our soleus muscle, so deep to the gastrocnemius, is super slow, all right? So they all get up to the same peak tension here on this chart. Because we gave it as a percent, your eye muscles are not as strong as your gastrocnemius. But in order to get to their peak tension, it took them different amounts of time. So that's partially due, right, to the difference in the type of myosin that we see there and how fast it can break down ATP, and therefore how fast we can do that cross-bridge cycle. Questions about this before I move forward, because I don't know, it's a new concept. Okay. So we're going to be layering this together. We're eventually building to understanding like what a fast glycolytic fiber would be. So that's our fast versus slow part. Now we're thinking about the glycolytic versus oxidative part, which I kind of touched on already. So in addition to having fast fibers and slow fibers, we're also going to have glycolytic fibers and oxidative fibers. The glycolytic fibers are the ones that are going to do glycolysis. So they're specialized for anaerobic glycolysis. So breaking down glucose, getting pyruvate, turning that into lactate. So we have this buildup of lactic acid, but we also keep having enough NAD to continue running this glycolysis. Okay. This means that they don't really need mitochondria, right? They're mainly doing glycolysis, which occurs out in the cytoplasm. So if we compare glycolytic fibers, so our glycolytic muscle cells, to oxidative fibers, in a glycolytic fiber, we would see fewer mitochondria. We would see a bunch more glycolytic enzymes. So the enzymes necessary for glycolysis, if we're doing lots of glycolysis, if that's the main way we're getting energy, we're going to want those enzymes. We also see high glycogen stores in glycolytic fibers. So we're quickly kind of pulling apart those glucose molecules so that we continually have supplies of glucose to break down to get our little 2 ATP. And our glycolytic fibers are also going to be pretty large in diameter. So they're going to be large in diameter. Basically, because one of the reasons for having a thin muscle fiber is so that you have a lot of surface area to volume, so that you can get a bunch of oxygen into the mitochondria. But if you don't need oxygen, you can build a bigger fiber, one that's thicker in diameter, which actually also means that you're able to have like more layers of actin and myosin, so more myofibrils, which is what makes these stronger, basically, our glycolytic fibers. The downside is that they're quick to fatigue, right? So they're not using oxygen, they're doing this kind of inefficient process to make their ATP. So even though these are going to be strong fibers, they're going to run out of energy quickly. So the opposite are oxidative fibers. So our oxidative fibers, their goal is to do oxidative phosphorylation. So they'll do glycolysis, but they're pulling that pyruvate into the mitochondria. They're doing the Krebs cycle. They're doing the electron transport chain. They're doing oxidative phosphorylation. Okay. They have lots of mitochondria for this. So oxidative phosphorylation happens in the mitochondria. So we need mitochondria. They also have this molecule called myoglobin. So myoglobin is kind of similar to hemoglobin. And we've talked about hemoglobin a little bit in lab, and we'll talk about hemoglobin a little more next semester. But the basic thing about myoglobin is that it stores oxygen. So it's going to be in our cells. Hemoglobin we'd see out in the blood. But myoglobin in our muscle cells stores oxygen. And just like the blood, it's red. So it's keeping that oxygen as in kind of this storage molecule. It's red. And this is going to give our oxidative fibers a different color. They're going to look different. So this is actually why there's a difference between white meat and dark meat. Like in the turkey we may or may not be eating next week. That dark meat is because of the myoglobin, because we have oxidative fibers there, because in the legs of the turkey, right? Turkey's, kind of running around all day. We don't want the legs to run out after 5 minutes. So we have these oxidative fibers that are slow to fatigue, keep going all day. So we need this myoglobin to store oxygen. Okay. Oxidative fibers, they need oxygen, so they also are going to be really well vascularized. They're going to have lots of capillaries going to them. And they're going to have lots of capillaries going to them to bring the oxygen. And then they're also going to be small in diameter, so that oxygen can diffuse easily into the cell, and then also diffuse easily throughout the cell to get to those mitochondria. Our oxidative fibers are pretty resistant to fatigue, so they can go on for a long time, so long as we have oxygen, which they're getting in 2 ways. They're getting that oxygen directly from the capillaries. They can also use this oxygen that they've stored in myoglobin. So while they're waiting for the lungs to kind of kick into gear, right, for you to start breathing a little harder, for your heart to start beating, they can be using this oxygen in the myoglobin to kind of top themselves off. Okay. So then we bring these 2 things together, right? The slow versus fast, and the oxidative versus glycolytic. So we're bringing them together, and when we want to name a specific muscle fiber, we're going to talk about it as either slow oxidative, fast oxidative, or fast glycolytic. So a helpful thing to notice here, right, is that all glycolytic fibers are fast. I kind of remember that by remembering that glycolysis is the first step, so you would get there fast. That's not why they're called fast twitch fibers, but it's helpful when you're trying to remember the names. Okay. So we're going to look at each of these in turn. Starting with our slow oxidative fibers. So they have this slow twitch, means they have this slow form of myosin, and they're oxidative, which means they're trying to do oxidative phosphorylation. They're trying to use oxygen. Okay. So we have this low myosin ATPase activity. So basically this myosin that breaks down ATP kind of slowly. And they have a high oxidative capacity. So that's saying that they're using oxygen to do oxidative phosphorylation. So this is going to be for aerobic activity. We're doing aerobic forms of creating ATP. So aerobic means using oxygen. This means that they have mitochondria, lots of them. They have lots of blood supply, right? They need that oxygen. They have myoglobin, so they're red. All right. As we just said, with our oxidative fibers, with all of our oxidative fibers, right, they're going to be small in diameter. So this really means two things, right? We just talked about this diffusion barrier, right? They're small in diameter, which means they have a relatively large surface area to their volume. But also because they're small in diameter, they can't have too many actin and myosin in parallel, right? Because they're kind of thin, which means they're not going to be able to generate as much force. So they're not going to produce as much tension when they contract. So that's kind of the trade-off here. So that we can produce lots of energy, but we can't contract so forcefully because we need to keep these fibers thin in order to keep them oxygenated. So they're going to fatigue slowly because they're good at making ATP. They're good at keeping that up, but they're not necessarily going to be able to produce big, powerful bursts. Our next type of fiber are fast glycolytic fibers. So the fast part, so they have this really fast form of myosin. So they have high myosin ATPase activity. So they're cross-bridge cycling quickly because they can break down that ATP quickly. Their myosin is able to get back into that high energy state, ready to bind actin quickly. The glycolytic part is referring to that fact that they're really mainly relying on glycolysis for creating ATP. They have high glycogen stores for this, as we discussed with glycolytic fibers, and they have lots of enzymes that are related to glycolysis. They don't need oxygen, so they also don't need myoglobin. So they're going to appear white. They're not going to have this red color. And they're large in diameter because they don't need to keep that big surface area to volume ratio. So they can get wider, which means they can stack more actin and myosin, which means they can be stronger. Con is that they're going to fatigue rapidly. They're not using a super efficient form of producing energy. So they're going to run out, and the muscles are going to start to burn as we build up lactic acid because we're doing this anaerobic glycolysis. So our final type are fast oxidative fibers. So fast oxidative fibers, they actually have intermediate myosin ATPase activity. So like they actually have a different kind of myosin that's kind of medium levels of speed, but they're fast compared to our slow oxidative fibers. They have a high oxidative capacity, just like our slow oxidative fibers. They have myoglobin because they still need that oxygen. They're slow to fatigue, but they're still going to fatigue more quickly than our slow oxidative fibers. They're intermediate in diameter. So they're in the middle on basically everything. Okay. So we have a nice chart for people who like charts, right? So this is comparing our slow and fast oxidative fibers versus our fast glycolytic fibers, telling you the color at the top, which tells you whether or not they have myoglobin. Red, yes. White, no. And then we can just look, what's their oxidative capacity? So are they doing oxidative phosphorylation? What's their glycolytic capacity? How fast are they contracting, which is related to how fast they can break down ATP. Talk about the mitochondria, right? We need mitochondria for oxidative phosphorylation. So that's basically just going to be the same. So most of these things are really related to each other. Yeah. The cool thing about fiber types is that they respond to exercise, and you can actually convert between fiber types depending on training. Okay. So every muscle in your body is a mixture of fiber types, and some of that mixture is going to be dependent upon genetics, specifically some of the like fast versus slow stuff. But you can convert some fibers. Oh, we're not to the converting part yet. That's clearly the part I'm excited about. Okay, the proportions in any given muscle will vary also on the function. So if we're talking about like your intrinsic muscles around your spine that keep you upright when you're sitting and standing, those have more slow oxidative fibers. You have to use them all the time. Whereas muscles that you just use some of the time, they can have glycolytic fibers, they can have fast glycolytic fibers, right? In a single motor unit, all of the muscle fibers are going to be the same type. So this means that we're actually, because we recruit our motor units in a sequence, this means that we also recruit these different types of fibers in a sequence. So this is why I was talking about that sequence in the beginning. Okay, so our recruitment order is going to start with our slow oxidative fibers. So our slow oxidative fibers are small, so we're recruiting them first. Then we're going to recruit our fast oxidative fibers. So they're still oxidative, but they've got this faster form of myosin, so they're kind of intermediate. So they're coming second. And then we're going to recruit our fast glycolytic fibers. Those are going to be our big strong ones. We're going to do this when we're sprinting or when we're weightlifting. Okay. So we're going to recruit them in an order based on kind of like building up how much force we need to produce. So our fibers have different resistance to fatigue, as you could tell from the bulleted lists that talked about fatigue on each of them. But when we're talking about different activities, we can think about why different activities make us fatigue more quickly. So with high intensity exercises, we're using those glycolytic fibers, or fast glycolytic fibers, because they're bigger in diameter, so they have more myofibrils, so they can produce greater tension, greater force. This means we're building up lactate, right? Because that's what happens when we do anaerobic glycolysis, which means our muscles will start to burn. So that's one thing. The other thing that'll happen as we're doing this high intensity exercise is that as you contract these muscles, right, when you contract them really forcefully, they're bunching up and they take up physical space, which means that they can cause compression of blood vessels. So as we're compressing blood vessels, we're actually kind of stopping some oxygen flow to the muscles. So this is going to impair any oxidative phosphorylation that might be going on. We also have neuromuscular fatigue. So as we're sending rapid action potentials down our motor neuron, we can actually run out of acetylcholine for a little while. So you can actually, basically block this from that neuromuscular junction side. We'll also have altered enzyme activity in these fibers. We'll be able to recover within minutes to hours because this is really about that buildup that we can clear, right? Once we relax this contraction, our blood flow is going to return. You don't run out of acetylcholine for that long. So if you take a rest, everything will go back to normal pretty quickly in terms of your ability to produce energy again. We're going to contrast this to our low intensity exercises. Even though they're not necessarily as hard as while you're doing them, right? They're lower in intensity, right? By definition, we're using that word. If you actually keep going until you really deplete your energy, it's going to take a long time to recover because one of the things that you're depleting with low intensity exercises is glycogen. Okay. Honestly, they understand a little bit less about how this works, is my impression. They understand very little, at least from a biology perspective, on how willpower plays into this, but fatigue also does have a mental component, right? There's this will to win if you're in a race, and that does also affect how quickly you fatigue. Maybe ask your psych professors about that one. Okay. So here we're looking at the muscle fatigue. So we seen charts something like this before. Maybe we saw some in lab, but we also saw some as we were talking about like tetanus in our previous unit for all of 10 seconds. But what we're seeing here is the pacing of stimuli, right? So each of these arrows is a stimulus, and we're looking at the tension that this stimulus, that this action potential is producing. So here we see we are stimulating our muscle, spaced out, giving our muscle time to relax between these stimuli. We can keep producing that same amount of tension for a long time. All right. As we start to send faster action potentials down our motor neuron, our stimuli are getting closer together. We produce tension for a while, but near the end, we start to fatigue. So we start to experience these things we're talking about here. Yeah. Finally, when we're sending stimuli really quickly, we may reach this state of tetanus where we no longer see gaps between our tension at all. We're not relaxing at all. And then we'll see fatigue. And if this happens after a rest, we'll be able to get back up to that peak tension again, but we're going to tire much more quickly because we haven't rebuilt our stores of everything that we need. So this is what I wanted to get to. So we do see long-term muscle responses to exercise, right? One of the reasons people exercise, right, is they train. If you're an athlete, you train to do your sport. So different types of activity are going to have different effects on our fibers. So with aerobic exercises, we're going to see increased oxidative capacity. So we're going to be increasing our ability to do oxidative phosphorylation. We can actually convert some of our fast glycolytic fibers, so those ones with the faster form of myosin, that are doing mainly anaerobic glycolysis. As we're increasing our oxidative capacity, we can actually convert them to fast oxidative fibers. I think that's super cool. So we can like increase their numbers of mitochondria, right? We can do all sorts of things to them that make them more immune to fatigue, right? So we're increasing the size and number of the mitochondria. We would decrease the fiber diameter as we do this so that we can increase the number of capillaries so that we can get oxygen to these fibers so that they can switch over to doing oxidative phosphorylation so that they can give us long endurance for these activities. For high intensity exercises, so here we mean like weightlifting or short but very fast sprinting as opposed to a marathon, right? We're going to see decreased oxidative capacity. We're going to see some fast oxidative fibers converted to fast glycolytic fibers. So that's the opposite of this one, right? Notice that we're just converting from fast to fast. We don't see any slow to fast because it's dependent on the form of myosin that we have, which is harder to change than like what organelles we have. Okay, so we can convert some of those fast oxidative fibers that were more built for endurance before, and we can convert them to fast glycolytic fibers that are now going to have more layers of actin and myosin, so we can then kind of adapt them for strength. As we do this, we would decrease the size and number of mitochondria. We'd be increasing the fiber diameter because we don't need as much oxygen anymore, so we can form more myofibrils. And we're going to see reduced resistance to fatigue, so these fibers are going to get basically stronger, better at producing tension, but easier to wear out. We do also see changes to motor fibers or muscle fibers in response to atrophy. So if you don't use them, they are going to go away. So the fibers will decrease in size. You may have noticed that muscles in the cadaver lab, right, are different sizes between different cadavers, right? So we do see real differences in the size of the muscle, and that has to do with these differences in the size of the fibers. So we'll lose myofibrils if we don't use those muscle fibers. And this will happen in some disease states as well, right? So this isn't just from being a couch potato. This also happens in things like muscular dystrophy. So this is just crossing that objective out so that you know you don't need to know it. We're not going to be talking about how we're controlling the skeletal muscle activity because we talked about antagonistic muscles in lab, we talked about reflexes there. So we've at least touched on that subject. So we're going to move on real quick to smooth and cardiac muscle structure. I think the slides in here are just on smooth, and then we'll talk about cardiac once we're actually in the cardiovascular unit. Okay. So we just want to know a couple quick sort of trivia facts about smooth muscle and how it differs from skeletal muscle, which we've spent a lot of time on. So what we've been talking about thus far is really just skeletal muscle. So we've been talking about the somatic nervous system. We've been talking about those neuromuscular junctions with their acetylcholine. Even the sarcomeres that we've been talking about, we're not going to see them in smooth muscle. There are tons of differences in smooth muscle. And if you look in your book, they'll talk about even more of them in detail. More differences in excitation, contraction, coupling, works totally differently with different molecules. We're not going to go that in depth, but we do want to know a little bit about how smooth muscle is different, because we are going to be talking about some smooth muscle as we talk about blood vessels. Okay. So if we're looking at smooth muscle, we're going to see that it lacks striations, so it's not striped. That's because it doesn't have sarcomeres. Okay, so we don't have these structures with their Z lines and their M lines and their bands. That's not the structure of a smooth muscle. The smooth muscle cells are going to be kind of teardrop shaped, right? And they're going to be kind of staggered. They're not in these big, long motor fibers. Okay. We're going to find them in different places. So they're not in our named skeletal muscles attached to bone. They're found in internal organs and blood vessels. And they're under involuntary control. So you're not thinking about this and contracting your skeletal muscle. This is happening as an intrinsic process in response to some sensor in your body. We'll talk specifically what sensory receptors those would be later. So they're spindle shaped, they're small. They still contain actin and myosin. Said they don't have sarcomeres. They do have something called a dense body. That's a specific structure. A dense body just kind of communicates what's happening between the actin and myosin to the outside of this muscle fiber. But basically how they're contracting, right, because they don't have sarcomeres, is that they still have actin and myosin. They still have these thin and thick filaments, but they're arranged kind of obliquely, and they're actually a fair bit longer. So it looks kind of like this network kind of crisscrossing around our smooth muscle cell. And the dense body is kind of anchoring that to the surface so that as they slide past each other, they still slide, right? They just kind of slide at these angles. They're still doing cross-bridge cycling. This dense body connects it and makes sure that the smooth muscle cell as a whole shrinks. Okay. Smooth muscle is also much slower than our skeletal muscle. So we still have the sliding filaments. They have myosin heads along the whole length because we don't have the sarcomere, so we don't have this specific bare zone in the middle. We just kind of have overlapping the whole range, longer range of contraction. You know what, we do have a fair few number of slides left, so we'll touch on these last couple of things. But if you want to preview, the main thing I want you to know about smooth muscle from the next ones is that they have gap junctions, and their junctions between the neuron and the cell look different. We see those neuroeffector junctions that we talked about briefly with our sympathetics and parasympathetics here instead of our neuromuscular junctions. But I will come back to that. I'm sorry, I know I'm over time. Okay. Have a great weekend, everyone.